All right, what about the brain? Well, here's a picture of a brain, and here's our basics of the visual system. We've got the eye here. In the back of the eye are about 100 million photoreceptors in each eye, about 100 million. Those converge down onto about 1 million ganglion cells in each eye. These ganglion cells have very long axons. They send their axons all the way back. They are the optic nerve. That's what the optic nerve is. It's ganglion cell axons. The ganglion cells at the back of the retina. Actually, it's the front of the retina. You know, your photoreceptors are at the back of the retina, and the light has to get through the ganglion cells to get to the photoreceptors. It's kind of a backwards way to construct an eye, but that's how the eye is constructed. Ganglion cells here, photoreceptors behind them, and then the ganglion cells send all their axons out through the blind spot. You have to puncture the photoreceptor layer to get out of the eye. You know that squids do not have a blind spot? Their eyes got built the other way around. Right? So they have the photoreceptors in front and the ganglion cells behind. They should be truck drivers. Okay. Thank you. All right. Then we got this little way station, lateral geniculate body. It's always thought of as kind of a relay station for these ganglion cell axons, but in fact, only 10% of the input to this lateral geniculate body comes from the eyes. The rest, 90%, comes from the brain, and we're not really sure, well, it comes from the cortex and other structures in the brain. We're not really sure exactly what all that 90% of inputs are doing here, but they're obviously modulating the input from the eye somehow. Visual attention, helping you find what's red in the scene that you're looking at, something like that. And then you have these optic radiations heading off back to area V1, also called primary visual cortex. Area V1 or primary visual cortex, that's the first site at which the input from both eyes is combined. That's the first binocular area, and it's the first place where you see really, really good motion detectors. And we're going to be taking advantage of your V1 cells, your primary visual cortex cells this evening. It's actually, this is the right, somebody's right hemisphere. We're looking at it in the middle, right? So we've cut down a sagittal slice through the brain here. We've cut the corpus callosum in half. Here you can see the cerebellum, and this little fold here is the calcarine sulcus. Almost everybody has a calcarine sulcus. All the rest of this differs a lot from person to person, but this is pretty standard. Some of these other sulci are also pretty standard. Just a cross-section, stained, so you can see the gray matter and the white matter. And here you can see in primary visual cortex there's this white stripe. That's why it's called striate cortex, in addition to being primary visual cortex in V1. Striate cortex. There's actually a layer of axons in that part of cortex that carry information back and forth from one part to another that you don't find in the rest of cortex, like the frontal cortex. Okay. And then there's one more important part for our purposes. There's a little spot of activity. It's driven by area V1 and several other areas. This area has neurons, lots of them, that really like motion and really like binocular disparities. And you see it light up. If you do a functional magnetic resonance image and you show somebody patterns that are moving while they're sitting in an MRI scanner, you'll see that little spot light up beautifully for moving things and also for stereo things. Okay, so areas V1 and MT both are rich in neurons that respond to motion and to binocular disparity.